Hello everyone, I am Sarita Rawat, Assistant Professor, School of Pharmacy, Suresh Gyan Vihar University, Jaipur, Rajasthan. Today we will start our next lecture of pharmaceutical microbiology. Today's our topic is fungi. So, learning objective of today's lecture is to study about fungi, to study about medical significance of fungi, to study about various type of fungi. Now, learning outcomes of today's lecture included fungi, importance of fungi, various type of fungi, medical significance of fungi. So, we'll start with a basic introduction. As you know, in microbiology, we have virus, algae, and bacteria, fungi. So, as you can see, for example, viruses, we have Ebola virus, then we have algae, algal bloom. So, you can see the picture very clearly. Then we have bacteria, Salmonella, and then we have fungi, for example, mushrooms. So, these microorganisms are our friend and our enemy too. Some of them cause disease and some of them are very helpful in our day-to-day life. Like for example, mushroom. So mushroom is very common household thing. So you ever eaten the vegetable of mushroom, and then the algae is also very helpful. And if we talk about our enemies, so bacteria like salmonella, virus, ebola, so they can cause very severe disease and they are very harmful for humans. Now, the variety of microbes we have categorized them as cellular, that are virus and prions. They are prokaryotic, cellular without nucleus, and we have bacteria, archaea, cyanobacteria, and then we have eukaryotic, which are cellular with the nucleus, that we have algae, fungi, and protozoa. So today's our lecture is fungi, and the fungi is having a cellular with nucleus. So if we categorize the microbes, so you can see acellular and cellular. So cellular, virus, prions, virus. And if we talk about the cellular, so we have prokaryotic and eukaryotic. Prokaryotic included archaea, bacteria, and cyanobacteria. Then in eukaryotic we have algae, fungi, and protozoa. So today's our lecture is all about fungi. So fungi, as you can see, we will cover structure. What is the structure of fungi? Then cell. What is the cell of fungi? And shape, size. Then the reproduction of fungi, classification like yeast, mold, fleshy fungi, and then medical significance of fungi. Then summary. So, as you can see in this picture, we have bread and very other eatable things. So these like product include fungi to prepare them. So basically we can see fungi are too important for human food also. Like study of fungi. So mycologist is study about the fungi, we called it mycologist. And everywhere saprophytic in water and soil, and parasitic or in animal and plants, and harmful, they are harmful, unpleasant, fungal disease, spoilage of food, and beneficial is food and drinks and drugs. So as you can see, they are also used in some drugs and some food and drinks also. And no plant, no photosynthesis. We have a saprophytes. So as you can see in this picture, so saprophytes are the microorganisms, those who are able to decompose the garbage. So main source of food and that decaying organic material, then decomposition material into the absorbable nutrients, we call them recyclers also. No fungi means endless piles of the plant and animal waste. So as you can see, we have very large amount of garbage near us, and like in our environment where we dump our household things, our various like waste material. So fungi are very important to recycle them. They are very important to like they decay them, and they make them very clear, or they recycle them. We can say in one word, they are the recycler of the environment. So as you can see, the cell of fungi is they are a eukaryotic type cells. So fungi having eukaryotic cells. So it involves cell envelope, which involves cell membrane, cell wall, and as you can see in this picture also, we have the eukaryotic cell of fungi, and this is it also include the nucleus with the chromosomes and cytoplasm, endoplasmic reticulum, ribosomes, Golgi complex, mitochondria, and flagella. Flagella is very rare, but it is there in some fungi cells. Then the difference between prokaryotics is 10x larger than the bacterial cells contain two nuclei. Okay, so this is the structure of fungal cell, and it includes the mitochondria, Golgi apparatus, microbodies, ribosomes, nucleus, outer envelope, endoplasmic reticulum, and cytoplasm. So when we talk about all the part in detail, we see the cell envelope includes cell membrane. Okay, so cell membrane is flexible thin skin that enclosed the cell interior and controls in out transport of substance by protein in the membrane. So as you can see, this is the membrane protein, and they control the in out transport of the substance, and this is the cell membrane, and this is the beta glucans, and this is the chitin. Okay, then if we talk about the cell wall, okay, so it provides rigidity, strength, and protection to the cell. So it is very rigid, made up of very rigid material, so that the cell, the cell will be protected. It is much simpler in structure than bacterial cell wall. Okay, and cell wall of fungi contain polysaccharide chitin, we call it chitin, and also found in exoskeletons of crabs. So as you can see, what is present in the exoskeleton of the crabs, that is similar thing present in the fungal cell wall, and that is made up of polysaccharide, so which make it very strong. So when we talk about the nucleus, so we find out primary difference between prokaryotics and eukaryotics is command center control the function of entire cell. So as you can see, if we talk about the prokaryotic and eukaryotic, so the common difference between prokaryotic and eukaryotic cells is eukaryotic cells having nucleus in their cell, and the command center of the cell is nucleus, so it controls the function of entire cell. And there is three components of nucleus, that is nucleoplasm, that we call matrix also. It has chromosomes, that is a linear DNA molecule, genes, genetic information, which include the genetic information which passes through the genes to gene. And then it also includes the protein and nucleus membrane with pores. Okay, so as you can see, we have cytoplasm. So if we talk about the cytoplasm, it has semi-fluid matrix with the cytoplasmic organelles having specific functions like endoplasmic reticulum, that we call ER. It is a surface for ribosomes to attach, And then what is ribosomes? Ribosome sites for the protein synthesis. Then we have a Golgi complex. Golgi complex help in packaging of the plant transformation, synthesize protein into the functional one and the package them. Then we have mitochondria. So they are powerhouse of the plant. So a powerhouse of the cell. So you have ever heard in your childhood also that mitochondria is the powerhouse of the cell. So it is a power plant produce energy to drive cellular function. Okay, so all the power required to function the cell. Okay, so the energy required is all taken from the mitochondria. So as you can see, this is the cytoplasm. All the things is included in this. Then this is the structure of fungi. So as you can see, this is the unicellular yeast colony here, and this is the culture medium, and this is the filaments, hyphae, mycelium, mold colony, and this is the vegetative hyphae, as you can see, and this is the petri dish, and this is the cytoplasm divided site of okay, so this is the cytoplasm multinucleated. This has a septate hypha, and this has a aseptate hypha, and this is the sepitum, as you can see. Okay, so now if we talk about the size of fungi, okay, so size of fungi, it has a typical size. Yeast has diameter of 3 to 5 mm, 1 million microns or 1 meter. Then we have a separate hyphae width of the 2 to 15 mm, and it become very large in the length. Aseptic hyphae with width of 10 to 30 mm, which becomes very less in length. So these are the molds, yeast, and these are the bacteria. Then this is the microscopic appearance of fungi. Now reproduction in the fungi, how the reproduction occur in the fungi. Budding, by budding, so it is a asexual mode. Budding is a asexual mode. So bud grows on the parent cells and nucleus divided, and one of the them nuclei, and it pass into the daughter cell that called bud, and bud separated from the plant cells. And if we talk about the fragmentation, the next type of reproduction is the fragmentation. So it is also a asexual type, and in this hyphae is separated into the pieces, and each component grow as separate entity. So as you can see, this is the budding process, and this is the fragmentation process. Now the another process is formation of spores. Okay, so formation of spores is of two types, like sexual spores and asexual spores. So sexual spores included fusion of two nuclei, and asexual spores like in conidla, it has a no fusion of nuclei. So as you can see in this picture, this is a separate hyphae, and this is the conidia. So it is there is this in this fungi is having a asexual spores. Then the another type of reproduction of fungi is fungal spores. So it has very resistant structure and resistance to heat, cold, acid, base, etc. And they carry great distance by wind. Many people are allergic to the fungal spores. Then we have a microbial classification. So taxonomy, if we talk about the kingdom of fungi, so it has divided in 5 phyla. So as you can see, this is the kingdom of fungi. First one is we have it is selected with according to the characteristics of the phyla of the fungi, and we have categorized them into the 5 types. First we have food spoilage, then water moles and plant disease, mushrooms and penicillium. So these we have categorized them with phylum and type of hyphae, type of sexual spores and the type of asexual spores. So first one is food spoilage, that is zygomocyte, that we call zygomycota. Zygomycota is a accepted, and it has a zygospores, and it type of asexual spore. It has a nomitile sparangospores. And then we have a water mouths. Water mouths we have a Cytridomycota. Plant disease we have a psychomycotina. Then we have mushrooms. Mushrooms we have basidomycota. Then we have pelicillium, which we call deuteromycota. Okay, so these are the taxonomical division of the fungi kingdom. So three important types are, we have yeast, modes, fleshy fungi. If we talk about the yeast, okay, so yeast is a single cell. It is a unicellular and it has no hyphae. So it has an individual cell, like we have a blastospores and blastocandida. Okay, and in single cell unicellular yeast, the reproduction is caused by the budding method majority and spore formation method like least commonly. Okay, and the present, it is happen in the presence of soil, water, fruits and vegetable. Then the fermentation happened by Louis Pasteur by yeast. We discovered it. It has a anaerobic in the presence of sugar and alcohol. So it is used to make wine and beer. Then we have aerobes. Aerobes mean we have sugars in the presence of CO2 and water. So it is used in making of bread. Then we have pathogens. Pathogen we have candida. So as you can see, this is the yeast colony. This is the budding yeast. Then growth. So for the growth, we have an experiment. How can we see the growth of yeast? So we have taken is as an experiment. So topic is growth of yeast. So first what we have to do, mix the yeast and the sugar with some warm water and stir it in a bowl. Then pour the mixture into the bottle and cover the bottle with a balloon. Then put the bottle on the top of the heater and observe the balloon. Then repeat the procedure without adding sugar to the yeast. So what will you see? Then what will you see? What does yeast need to grow? The yeast need sugar in the source of energy, as we already discussed it in our previous slides. So what caused the balloon to blow? Yeast break down sugar to ethanol, that is alcohol, and the carbon dioxide. So process is known as fermentation. Production of bread, beer and wine is processed by the same process of fermentation. Carbon dioxide released from the fermentation process in filling the balloon. And when making bread, CO2 causes dough to raise to make a light spongy loaf. So as you can see, you have seen the bread in your household, in your kitchen. So the bread is like a very spongy material, and it is very floppy. So yeast make it like that. And in the absence of sugar, yeast lost its source of energy. No CO2 is produced. Now we talk about the modes. So modes are the filaments, hyphae, and mycelium. They are aerial hyphae and they are a vegetative hyphae. Their reproduction is caused by the spore formation, either by the sexually or either by the asexually. Then it is in the presence of soil, water, food, spoilage like bread molds, potato blights, and food flavor like cheese. And it is used in the production of antibiotic like penicillium, enzyme, and acids. So as you can see in this picture, so this is the mode that is commonly grow in your like if you take out the potato, okay, and you put it for some time, some days, so you can see some kind of blackish material there. So that is the mode that grow in the potato, and then also in the cheese. Then if you can if you have a bread, so you can see all the things also, like bread also is very productive source to grow the molds. So if you take the bread for like few days in the year, in the like in your kitchen or somewhere, so you can see this kind of things growing in your bread. So they are the modes. Then we have a fleshy fungi. So fleshy fungi are the large fungi that grow in the forest. So for example, we have mushrooms, and we have toadstools and puffballs and bucket fungi, and they are no microorganism. Then structure, they have a network of filaments that we call mycelium, and in soil or rooting log, they found in soil and rooting log, and fluttering body, they have spore formation and release, and spore germinates into new organisms. So the type of reproduction they follow like with the help of spores. Then they are delicious food and toxic to health. So as you can see, we have mushrooms that are very tasty and edible also, but at the same time we have some kind of mushrooms, those which are very toxic and very lethal to consume. So if any person consume it, they can lose their life also. So wherever we are consuming any kind of mushroom, so we have to check it, either it is edible or it is very lethal. Then what is the medical significance of fungi? So as you can see, fungi is very important for making of antibiotics. Like fungi that cause disease are pathogens, plant, mold, blights or rust. So human animal inhalation bound, microbial in toxin, mycotoxin, and it infectious disease also like mycosis. So if we see the fungi, so they can also cause disease, some kind of fungal disease, like this is the example of some fungal disease. Like you can see, yeast can cause the C albinica, so which they can cause a disease like thrush and yeast vegetative anal infections, systemic infections. Then mold also cause aspergillus sepsis by they can cause the systemic infection. So these are the example of some fungal disease. Okay, now as you can see, myos can cause superficial infection. Superficial infection means the upper layer of your skin, like in your hair, nails, in your dead skin layer. So it is a epidermis infection. So like toenail infection. This is the example of myos infection, superficial fungal infection. So they can cause the toenail infections. Then cutaneous infections, it can caused by living layer of skin, dermis. So they can cause it in the living layer of the skin, like in your lips. For example, this is the picture: mouth, skin, vagina infection, and like candida yeast. Then it can also cause subcutaneous infections like in your dermis or underlying tissues like medulla fruit. So this is the medulla fruit. Medulla fruit is very dangerous. So as you can see, it can like spread over all over your dermis layer. So the cure is very important. Then the another is systemic. So systemic is internal organ and infection of the lung, bloodstream and nervous system. So fungi also can cause very severe disease. Then the diagnosis and treatment for fungal disease are first identification of yeast, then culturing of them by biochemical test, then identification of modes, microscopic observation, their color, texture, topography, their growth, then microscopic observation of their structure of the spores, and then the treatment. So antifungal agent, we have very antifungal agent to the treatment of the fungal disease, like we have niacin, and then we have amphotericin, then we have fluorocytosine, and then we have cutisin toxic. Then we have demographic fungi. As you can see, this is the demographic fungi. Demographism in vitro, it is in the 37 degrees Celsius yeast, and the in vitro it has 25 degrees Celsius more for moles. Then we have pathogenic skin disease and lung disease it can cause. That we have lynches. Next we have lynches. So as you can see, lynches mainly grow in the forest areas. It is colored like cellular patches. One tree trunks and rocks, they grow in the tree trunks and the rocks. They are in the gray, brown, black, orange and green color, and they have leaf like structure and crust like and shorobre and gelatinous structure. Then they have symbiotic relationship with algae and cyanobacteria, which we also call photobionts, and they are fungus. Then mucosilium, both parties benefits and antibacterial. They also have antibacterial properties. So as you can see, these are the mucosilium. So slime mounds. Next we have slime mounds. So slime mounds is the combination of fungi and protozoa. Like they have like you can see, this is the combination of fungi and protozoa that we call slime mounds. So they grow from the spores, and then they are germinating spores, then they are growing like this, and then you can see they are growing the spores while maturing, and they are the combination of fungi and protozoa. So as you can see, next we have summarized what is bacteria, what is virus and what is fungi. Only you can categorize the fungi when you know about the virus and bacteria, and we will go in deep about them. what is bacteria, what is virus, and what is fungi. So what is the common difference, so that we can classify the bacteria, virus, and fungi into different types. So if we see the cell structure of bacteria, so they have no nucleus, and they have a complex cell wall. Okay, they have no nucleus and no complex cell wall. Then depend on the host cell, the virus cell structure depend on the host cell. Then fungi is a true nucleus. Fungi has a true nucleus, and it has a simple cell wall, as compared to the bacteria. Bacteria has a complex cell wall. Then the reproduction in bacteria, there is a binary fission, or we can say bacteria has a asexual reproduction. Same virus has a asexual reproduction, but fungi has a budding, fragmentation, spore release. So they have sexual and asexual reproduction both. Then, if we talk about the structure of fungi, so we find they have a spherical structure, spiral structure, and in virus they have a spiral polyhedral structure, and they have helical structure. They have a complex structure also. In fungi, the structure is spherical, mainly oval type or filamentous type. Then if we talk about the size, so bacteria has 1 to 3 μm size, and virus has 0.01 to 0.3 μm size, and fungi has the biggest size 3 to 30 μm. Then study of bacteria is called bacteriology. Study of virus is called virology, and study of fungi is called mycology. Then it has a pathogenic, so all are the pathogenic. So this is the summary about the fungi. Now the growth. So we have taken a simple experiment to see the growth of the fungi in common day-to-day life. So this is the common, very simple experiment. You can also follow this in your home very easily. So first you have to see, we have a title that potatograms, and the topic we have a growth of bacteria. So we are checking the growth of bacteria. So we have already seen the growth of fungi in your bread, in your cheese, in your potato. Now we will see the growth of bacteria in the potato. So first, peel and slice the potato with gloves in 4 pieces of equal size. Put 1 slice with glove in box labeled control. Then put second slice with glove in box labeled oral, and cuff below on it. Then take third slide and rub it on the table before putting it in the box and label table. Then take out your gloves and rub fourth slice with your hand before putting it in box labeled hand. Now what you will find? So explain. You have to see all the differences. So this is your experiment for to do your homework, so you can check and differentiate between the what happened in different situation. Basically the aim of this experiment is to see the bacterial growth. Okay, so explain the difference in bacterial growth on the sample. So you will find when using gloves, the potato does not get contaminated with germs. Okay, then you will find therefore control show only few bacteria growth, and the contaminated sample show more bacterial growth. And at what other places apart from the human body bacteria can be found? Everywhere on the earth, in soil, in water, on planet, even in the radioactive wave. So everywhere is the bacteria present. So do all bacteria cause disease? No, some bacteria are even beneficial to us. So they can inhibit the growth of disease causing microorganisms like pathogen. So this is very simple exercise I have given to you, potato germs deducting. So what you can do, you can take some potato and you have to slice them first while you are using the gloves, and then you have to cut it into the four pieces, and the one of the piece you have to label as a control, and you have to take it in a box while using your glove. So what is our result comes with this? When using gloves, the potato does not get contaminated with germs because we have followed all the precautions. We have not allowed the germs to come into that potato slice. Then the second thing what we have done, put second slide with glove in the box labeled oral and cough below on it. So what happened in this? Therefore, the control show only few bacteria growth. Then the third slice are rubbed in the table before putting it in the glove. So what happened when we rubbed it? So some kind of bacteria like a stick with that, and the contaminated sample so the bacteria growth. Okay. Now take your gloves and rub the fourth slice your hand before putting it. Fourth, in soil, in water, or in plant, everywhere we have a bacteria and germs. And we have done two simple exercise in today lecture. You can definitely try it in your home so that you can see the growth. Now we have concluded some quiz questions related to these chapters, related to today's lecture. You can easily answer this. We have multiplied them into two sections. First is true and false, and then multiple choice question. So first one is fungi are like plant photosynthesis organisms. So is this true or false? And unlike any other microbe, the cell wall of the fungi are composed of chitin, and dimorphic microbes are a contamination of algae and fungi. Fermentation of yeast is used to produce blue cheese. Fungi can cause both endotoxins and infectious disease. So these are the five true and false. You can easily answer them. Then we have a multiple choice question. So multiple choice question included the release of an enveloped virus practice from a host cell is called. So we have three options: budding, lysis, and escape. Then, which statement about the replication of bacteriophage is false? So, first option is it takes approximately 30 minutes to replicate, and B option replication take place by a lytic cycle, and third option the bacteriophage enter the host cell. Then another question, reproduction of fungi can be asexual, sexual, or both sexual and asexual. So we have already studied about this. Then the sexual spores are produced by a process called conidilla budding, the fusion of nuclei. Then the most dangerous form of mycosis is systemic, superficial, or cutaneous. So you have to answer these kind of questions. You can easily answer these by going through the whole lecture. So today we have already studied about the fungi, how it grows, the replication, what is the example of fungi, how is the types of fungi we have, what is the reproduction in the fungi, what is the medical significance of the fungi. Now you can correlate it like various fungi is very like dangerous, or we can say it fungal disease can cause very lethal, nowadays the side effect of some medication is also cause fungal disease. Now if we talk about the importance importance of fungi in microbiology. So why we see the fungi, why we read the fungi in microbiology. So as you can see, fungi are useful also in making a various type of antibiotic. So when we take in all the knowledge about them, only then we are able to make, or we are able to study about them. Okay, so fungi is like group of eukaryotic organisms that include microorganisms such as yeast, molds, and they are as we know, we are more familiar with mushrooms. So you can correlate it. And then we have a four type of fungi. That is, we have a club fungi, we have yeast fungi, we have bread molds. Okay, and then if we talk about the characteristics of fungi, so fungi has a eukaryotic cell. They are eukaryotic, non-vascular, non-motile. They are heterotropic organisms. They are more unicellular, filamentous. Then they reproduce by the means of spores, budding. They exit in the phenomenon of alteration of generation, and they do not have chlorophyll, so they do not go through the photosynthesis process. And if we talk about the microbiology of the fungi, okay, so we can found that they are multicellular organ, filamentous organ. They are a microscopic filamentous fungi that form large they have a large fluttering bodies. Okay, and sometimes the group is referred as a mushrooms. And like mushroom is just the part of the fungus we see above the ground. Okay, we have very other varieties also that are not like seen. Then we have single cell microscopic yeast also. We have multicellular filamentous modes, microscopic filamentous fungi. Then yeast is very small. They are lemon shaped, single cell, and that are about the size of red blood cell. Okay, so this is all about your fungi. Now if we talk about the reference, so we have taken the reference upon the pharmaceutical microbiology Backwell significant publication, Oxford. And then we have taken some part from the pharmaceutical microbiology by Dr. N.K. Jain. Then some of the slides are taken from the microbiology slides here. So you can actually check out these references. Hope you take a good note from this fungi today's lecture. and I hope you understand this very well, and you can correlate this with your day to day life. So that's all for today. Till then, thank you for kindly listening to me. Thanks a lot.